am Dr. Amit Bhattacharya, Associate Professor at Department of Zoology, Ramjas College, University of Delhi. And today I will be talking about applications of antigen-antibody reactions in immunology and medical diagnostics. So we will be talking about how in an interesting way these antigen-antibody reactions or the interaction patterns have been used in the development of immunological and medical diagnostic kits. Now before moving into the antigen-antibody reactions or the processes, we should understand what exactly the antibodies are and how the specificity of the antibody reactions takes place. Now, what exactly the antibodies work for? So we all know about a term which is called as the immunity. So immunity is basically the state of protection from the infectious diseases, and this immunity is brought about by various cells and molecules which are functioning in our body. So the human immune system is broadly classified into two major categories. One is called as the innate, the other one is called as the adaptive component. So as we know, the immunity is the one which helps us to fight with the foreign particles in a more specific component way. Now the immune system has got both the specific component and not more specific or lesser specific components in this one. So one part is called as the innate immunity, which provides the first line of defense against infection, and the most specific component is the adaptive immunity, which challenge in a higher degree of specificity and which produces a very remarkable property of memory. So the memory cells are produced in this particular cases. Now the innate immunity, which is non-specific host defense, has been in the various types, like the anatomical barriers such as the skin, temperature. These acts as a non-specific host defense mechanism. So we see that whenever there is an infection or any kind of foreign particle invasion in the body, what happens is there is a rise in the body temperature. So this is brought about so that these microbes or these pathogens becomes inactive or can be killed by that. So this forms the first part of the innate immunity or the non-specific host defense mechanism. The second part is called as more specific, which is called as the adaptive immunity. So it is brought about by the lymphocytes and the antigen presenting cell, which presents the antigens in a more specified way by processing and presentation. Now the lymphocytes are the main types of the white blood cells which circulates in the body, and they are produced in the bone marrow by a process which is called as the hematopoiesis. Now the lymphocytes leave the bone marrow, circulate in the blood and the lymphatic system, and they reside in the various lymphoid organs. So in our body there are various lymphoid organs where these lymphocytes are present. So if you see the image over here, this image shows the various lymphocytes, the B cells, which has got the surface antibodies which are there, then the T helper cells which has got the receptors present, T cytotoxic cell which also have the receptor present on the surface of it. Now the cells of the immune system have been classified into various categories. So we know that in the blood there are red blood cells, then the platelets are there, and the leukocytes have been divided into neutrophils, lymphocytes, monocytes, eosinophils, basophils, like this way. So they all forms under the white blood cells category over there. Now if we see this particular image, these are the major lymphocytes which are present in the body, which are the B cells, which contains on the surface the antibodies, or which are the specific molecule which targets the specific antigen which is invading our body, the T helper cell and the T cytotoxic cell. So we can see the electron micrograph images also over here. Now the lymphocytes in the cells, the first category which comes are called as the B lymphocytes. Now the mature B lymphocytes are definitely, they are distinguished from their synthesis and display a membrane bound immunoglobulin which is more or less called as the antibody on its surface. So these acts as the receptor for the antigen, and it is approximately noted that about 1.5 into 10 to the power 6 molecules of antibody on the membranes of a single B cell is present. So they are identical binding site for a antigen. So these lymphocytes are the one in which contains the antibody on the surface of it, and these antibodies, when the invasion takes place, they are released and they go and target the specific antigenic epitope which is present on the antigen or the foreign particle. Now let's see what exactly the B lymphocytes, how exactly they function. So this is an image which is showing the antigen. So they can be foreign particles, viruses, bacteria, like this one, fungi. So as soon as these foreign particle enters in the body, what happens is these B cells get activated. And as these B cells gets activated, they produces certain plasma cells which are the antibody secreting plasma cell. So these antibody secreting plasma cell, they release the antibody, and these antibodies, they are specific for certain epitopes present on the surface of the antigens or the foreign particles. They go and attaches over there, and that's how they neutralize that particular foreign particle or the virus, bacteria from our body circulation. So these antibodies are very specific for a particular epitope because of a presence of specified region on its surface. Now we will come into the structure of the these antibodies or the immunoglobulins, how exactly it has been used for the development of the various immunological techniques and the medical diagnostic tools. Now the lymphocytes or the T lymphocytes also are a one type of lymphocytes or the lymphoid cells which are present in the body, and they have derived the name from the site of maturation, which is the thymus. Now like the B lymphocytes, these cells also have got a membrane receptors for the antigen. Now these T lymphocytes recognize the antigen only when they are bound to specific self molecule which are coded with a surface major histocompatibility complex or the MHC complex. So what happens is the antigen or the foreign particle which enters, they get into certain specific cells, they get processed and presented on the surface of the MHC molecule, and then only they are being recognized by this T helper cell or the T cytotoxic cell. Now with the help of the T cell receptor and the CD receptor molecules, CD4 and CD6 receptor molecules over here, that's how the reception or the location gets transmitted. Now this is the image which shows that how exactly an antigen, when it invades into the cell, it is processed and presented on the surface of the MHC, and once it is processed and presented on the surface of the MHC, they are being recognized by the T lymphocytes, and then subsequently there are certain cytokines and those things which are released which activate the T cytotoxic cell, and how these neutralization of these antigens takes place in the body. So all these important information have been utilized in the development of the various techniques and the kits over a period of time. Now this was a Nobel Prize which was awarded in the year 1972 for the discovery of the antibody structure, the chemical structure of the antibody. Now what exactly they said, that the antibodies or the immunoglobulins are basically the heterodimers. So there are 4 peptide chains which are present. 2 are the light chains, identical light chain, which has got a polypeptide of about 25,000 molecular weight, and there are 2 identical heavy chains which are larger polypeptides of molecular weight 50,000 or more. Each of the light chain is bound to a heavy chain by a disulfide bond, and such non-covalent interactions such as the salt linkages, hydrogen bond, hydrophobic bonds, it forms the heterodimer or the HL molecule. So we see that there are 2 light chains and 2 heavy chains, each of which are attached to one another with the help of the disulfide bond. We will see the structure of the antibody. So if we see the structure of the antibody, there are 2 light chains which are there, and in the center there are 2 heavy chains which are there, and the light chains are attached to the heavy chains with the help of the disulfide bond, and both the heavy chains are then attached to one another also by a disulfide bond which is present in the hinge region. So we can see there is a hinge region which is there, in which the disulfide bond linkage is there. So this is the basic structure of the antibody which is there. Now further, these antibody, if we see the first 110 or so amino acid of these amino terminal chain of the both light and heavy chain, it forms a specific region which is called as the variable region. So this highly variable sequence or the variable regions which is present both in the light chain, it is being said as VL, and in the heavy chain it is being said as the VH over there. Now the regions other than the variable region is called as the constant region or CL or CH region, which is present in this one. Now the amino terminal portion which is corresponding to the variable region is the one which is responsible for the binding of the antigen. Now this is the region which binds specifically to the specific epitope present on the antigen. So this particular property has been utilized in the development process. Now let's see the image over here, the structure of these particular immunoglobulin. So if we see over here in the immunoglobulin, there are two chains, one is light chain, the other one is the heavy chain, and each of these heavy chains are attached to one another with the help of the disulfide bond, and the light chains are attached to the heavy chain with the help of the disulfide bond. And on the amino terminal, if we see, there is a presence of a variable region which is there. So the variable region is present and the rest of the part is called as the constant region over there. Now that's how the property is being utilized. So the heavy chains of a given antibody molecule can be determined by the class of the antibody. Like in the IgM there is a mu, IgG there is a gamma, then IgA there is a alpha, IgD there is a delta, and IgE there is a epsilon. So depending upon these one, the 5 major categories of the immunoglobulin are being classified in the human immune system. Now this particular property of binding of the antibody to the antigen or the antigen determining region which is present on the antigen, more or less called as the epitope, has been used over here. So we can see over here there are two types of antibodies which are there. One is specific for certain antigen determinants and the other one is specific for the other type of the antigen. So they will bind specifically to that particular epitope or the antigen over there. So these particular property have been utilized further. Now this is again another image over here, how exactly these antigen antibonding reactions are being taking place. But what is interesting over here is, as this antigen antibody reaction takes place, there is a mesh like precipitation which is formed over there. This particular property is being utilized further in the development of the kits over here. Now the exquisite specificity of the antigen antibody interaction had led to the development of a variety of immunological assays which are basically based on the detection or the presence of specific antibody or antigen in these particular things. So these particular property have been utilized to develop certain immunoassay which help in the biological or the medical interest also. Now let's come into how exactly these interaction takes place. Now these interactions generally takes place with the help of the weak bonds, okay, weak bonds like the hydrogen bond, hydrophobic bonds interaction which is there. Now the interaction between the antigen and the antibody depends upon 4 major types of the non-covalent bond which are shown over here, like the hydrogen bonding which are there, the van der Waals force of interaction which is there. So these are the various interaction bonds which are present between the antigen and the antibody interaction. Now the combined strength of a non-covalent interaction between a single antigen binding site or the epitope on a antibody and a single epitope is called as the affinity. So we can see over here, when a single antigen binding site of an antibody and a single antigen binds, it is called as the affinity. When a multiple interaction takes place between a multivalent, which means that antibody which are specific to a large number of the epitopes over here, and the antigen, it is called as the avidity. So we can see over here the avidity is shown over here. Now the avidity of an antibody is better measure of the binding capacity within the biological system than the affinity of its individual binding sites. Now we come into the precipitation reactions over here. The monoclonal antibody which are shown over here, these monoclonal antibodies are binding to the specific epitopes over here. Now as the binding is taking place, we can see the epitope binding and the antibody binding over here. But in the case of the polyclonal antibody, we can see that the antibody is binding to one and more different types of epitope, forming a mesh-like or a precipitation reaction which is taking place. So in the case of the multivalent or the polyclonal antibody or the anti-serum, there is a formation of a precipitation reaction which takes place, and this particular reaction has been further utilized to form the various assays over here. Now what happens is, the antibody which are bivalent, a precipitation will not be formed with the monovalent Fab fragment. So Fab fragment is the fragment which is present in the amino terminal end of the antibody, which is specific to a particular antigen. Now with the help of this one, there is a precipitation curve which is formed. So the first part of the curve, if we see, is where the antibody number is higher than the antigen. Then comes a point where the antigen and the antibody number are equal, and there is a precipitation which is formed over here, and finally the number of the antigen increases. So this is a plot of the amount of the antibody precipitated versus the increasing number of the antigen concentration over here. Now the first these particular precipitation reaction in the gel, it yielded to certain precipitin lines over here. Now there are two types of the immunodiffusion reactions which were used to form the assays. One was called as the radial immunodiffusion method, which is called as the Mancini method, and the other one is called as the double immunodiffusion method or the Octoloni method. Now we'll come into each of them, how exactly the antibody-antigen interaction are taking place in this particular film. Now the first one is called as the radial immunodiffusion method or the Mancini method. In this, what happens in the agar plate, the antibody is present already in that particular agar plate, and in the center there is a well which is being created in which the antigen are being loaded. Now as these antigen are loaded, over a period of time they will diffuse in the agar, and what happens is, as the diffusion takes place, they diffuse to a certain point where there is a precipitin line which is formed, and this particular line, there is a formation of a precipitin circle, precipitin line, which shows that there is a ring which is formed over there. We will see some of the images over here. Now as this antigen, it diffuses in the agar plate, the regional equivalence where the number of the antigen antibody interaction is optimum, there is a formation of a precipitin ring which is formed, and this ring is formed over there. By comparing the area of this particular precipitin ring with the standard curve, we can tell the concentration of the antigen which is present in that particular sample. So it can be determined. Now if we see this particular picture over here, this is a picture where the precipitin ring are formed around the central well. So in the central well, the antigen was loaded, and as the precipitin ring was formed in the region of equivalence, we can easily determine the area of the region of equivalence, and then it can be compared with the standard curve. Now this was one of the first assay which uses this radial immunodiffusion method, which took place in the beta-2 microglobulin in the serum. We know that the beta-2 microglobulin concentration, it increases in certain kind of disease conditions like the renal diseases, the inflammatory diseases, the autoimmune diseases, AIDS, acquired immunodeficiency syndrome. So in the patient, if the patient sample, serum sample can be loaded onto the well, and on the agar plate we can easily see the diffusion which is taking place and the immunoprecipitin line, which can be then compared with the standard curve, and the concentration of that particular antigen can be determined from the standard curve. So this was one of the first paper which used the radial immunodiffusion method for assay of the beta 2 microglobulin in the serum. These particular antigen-antibody interactions are utilized in the various other processes also. Now we see over here how these antigen-antibody interactions are helping in our body system also. So in this particular case, these are some of the examples which we have already discussed, how it is doing the inflammation process, how it is neutralizing the various antigens which are present in the body. Now the second important method which was used by this antigen-antibody interaction was called as the double immunodiffusion method or the Ochterlony method. Now in the Ochterlony method, what happens is, instead of putting the antibody in the agar plate, what happened is both the antigen and antibody were loaded onto two different wells over there. Now as they moved or diffused across the well, they were loaded onto the well. So in the one well, if you see the image on the left hand side well, the antibody was loaded, and on the right side well, the antigen was uploaded over there. Now what happens is, over a period of time, they diffuse radially towards each other. Now what happens is, as we see, the antibody started diffusing from the left side of the well and the antigen started diffusing from the right side of the well. Now as it reached a region or equivalence present in between, what happens is there was a precipitin line which was formed, or the precipitation occurred over there. Now this particular method or the technique was utilized for the development of various kits over here. Now this is a Ochterlony double diffusion method which is there. So as the name is suggesting, double diffusion means both the antigen and the antibodies are diffusing over here. So what happens is, there will be formation of various precipitin line over here depending upon the antibody which is present specific for the antigen over there. Now there are various precipitin patterns which are formed in the Ochterlony method, like if the antibody is specific for that particular antigen, so there will be a identical precipitin line which is formed over there. But if there is a partial identity, then there will be a partial spur which is formed over there, because the antibody is partially specific to the antigen or the epitopes which is present in the central well, and if there is a non-identical, there will be a cross which will be formed over there, which easily shows that the antigen and the antibodies are not specific to each other. So these particular various pattern forms were utilized for the determination of the antigen-antibody interaction. Now this particular antigen-antibody interaction patterns were further utilized for the development of the immunoelectrophoresis assay, which combines the electrophoresis along with the double immunodiffusion method. Now the antigen preparation is first electrophorese. So in this particular process, what happens is, the antigen preparation is first electrophorese. We know that there is a process which is called as the electrophoresis. So what happens is, the antigen, based on their charge density or the charge, they diffuses in the well. So they diffuses to the various portion in the well over there. Now this particular method we will discuss in the next part of the lecture. So in this particular method, it also utilizes the antigen-antibody interaction pattern along with the various methods which we just discussed, like the radial immunodiffusion method, the double immunodiffusion method, and in the next part of the lecture we will take up some of the more interesting examples, how exactly these antibody interaction are taking place and how these particular ideas are being taken in the development of the various kits and the various immunological techniques over there. So with this note, I come to the end of the first part of my lecture. Thank you very much.